Welcome to reproduction and genetics. Now, I want to go back just a little bit before I jump into reproduction and genetics, because the thing about biology is that everything is connected, right? So we started out talking about the rules about life, which kind of gave you an overview. Then we talked about chemistry, about carbon and hydrogen and how they all interact, and how they work together. We talked about macromolecules, and some of these macromolecules we're going to encounter this time again. The macromolecule we're going to talk about in this particular unit is DNA. DNA is the blueprint of what you are, but DNA still is a molecule. So everything in life, biology, is based on physical structures, things that are real, things that exist. And so when you encounter somebody who says, "Oh, this magical drug that I have, it will cure all the cancers in the world," the question you should be asking them is, how exactly does that work on a molecular basis? And I want you to kind of think that I want you to think about the fact that everything has a basis in a physical, molecular kind of structure. Without it, it doesn't work. And so this is where reproduction and genetics comes in. So reproduction and genetics, the things we're going to talk about here, is we're going to first look at the chromosomes, which are a reflection of the physical molecule DNA. Um, and then we're going to talk about Mendelian genetics. Both of those things were discovered independently of one another. Mendel actually discovered Mendelian genetics first, and he discovered it by math. He didn't have microscopes. He didn't know he didn't know about chromosomes. And then the chromosomes were discovered later, and it turned out that the chromosomes explain Mendelian genetics. Scientists love when these things work out. Um, then more we knew about DNA, DNA is the blueprint. It's a form of it's a set of molecules, but your body isn't made from DNA. DNA is just inside of your nucleus, as we know from looking at cells, right? What your body is made up is mostly protein. So chapter 10 talks to you about how do we get from DNA to protein? What does the body have to do in order to turn the blueprint into the thing that makes you? And the thing that makes you, the protein, and the DNA that controls it, um, it also has to be controlled in some way, shape, or form. So you're growing for a certain period of time, and then you stop growing. It's very important to stop growing when you need to stop growing. If you don't stop growing, if you don't stop reproducing when you're supposed to be, stopping reproducing, then you get things like cancer. A lot of diseases are diseases of control of information. And so we know a lot about how genes are controlled from bacteria because they're a lot simpler than we are. So that's basically the big picture of what we're going to try to accomplish in this unit, in this module. So read chapters 8, 9, 10, and 11. Make sure you answer the sample questions for yourself. Use Mastering Biology in order to do that. Then we're going to get to, and as usually, you've got helpful materials here. You've got a tour guide for the chapters. There's also a folder for assignment materials. There's a DNA isolation procedure. And then there's the who's the victim blood type assignment. I'm going to talk a little more about this assignment in just a second. This is how DNA replication works. Watch this if you have an interest in trying to see this a little more closely. There are a lot of demonstrations this time. I want to point mitosis and meiosis are key concepts we're going to talk about this time. And one of the things I think students struggle with is that mitosis and meiosis, how they're different and how they're the same. Mitosis creates an identical copy. In humans, that means that we're using mitosis mostly for repair and growth, that kind of thing. Meiosis or meiosis is to create your germ cells, so the reproductive cells, because it creates a cell that has half the genetic material, because those cells then fuse again in order to make a new organism. Um, Punnett squares, students are scared of Punnett squares. I'm not really sure why. Punnett squares are magical. Um, then here, a lot of people always ask me where this video is at. This is associated with this goes along with the Who's the Daddy assignment. And this is a real story that actually happened to me. I was actually asked this question by a friend, and so I want you to think about that. Um, Protein synthesis, I like Amoeba Sisters. They do a really nice job explaining things. Um, and then gene regulation by Paul Anderson. Ken Burb's film is probably a little too much. It's an interesting film, but I don't want to make you watch it. So there's no nothing associated with it. Um, but then the next thing I'm talking about is GMOs, because one of the things that we need to talk about when or we need to think about when we think about um, genetics is we discuss GMOs a lot, and people, especially in Europe, are afraid of GMOs, and you've got to know what they are and what their dangers are or what their dangers aren't. Um, by learning about what they really are. So don't let people make you afraid of things you don't know anything about. Knowledge is power. And with knowledge, you can question people who try to make you afraid of things. It's really easy to manipulate people with fear. Fear of the unknown is a huge problem. And people weaponize the fear of the unknown to trick you into buying things you really don't need or being afraid of things that you really don't need to be afraid of. So read up on these things, read up on GMOs, and think about whether you should be afraid of them or not, and what the issues are with them and what the issues aren't with them. Sometimes there are issues with things that aren't obvious initially if you don't know enough about them. So that's that. Then we come to the assignments. And the assignments, we've got two parts again, because I don't want you to do the second part before you do the first part. So do the first part first. That's where the knowledge comes in. And in the second part, that's where the application comes in. So chapter 8 and 9, 10 and 11, mastering and quizzes. Isolate DNA and post the results to the discussion board. This is just supposed to be a hands-on kind of thing. Do it with your children. If you don't have the materials, look at the ingredients and see what you could use instead. During COVID, I did not have isopropyl alcohol, but I had hand sanitizer sitting around. And I thought to myself, well, wonder if it works with hand sanitizer. It turns out it works just the same with hand sanitizer. The experiment works better with strawberries than it does with spit, but it works with spit. So play with it. You don't have to use the exact things that I'm using. Play with it. See if you can, come up with a way of trying to make this happen without it. And if it doesn't work, then you've done an experiment and it didn't work. Um, Post that. says, hey, say, I thought that this was work, and it didn't for me. Um, the Punnett squares, this time the study notes are going to be very specific. I want you to practice Punnett squares. Um, and then this practice of Punnett squares will help you tremendously for the assignments part 2. Here's your DNA isolation board, where you just need to answer some, put your pictures, and talk a little bit about what you did. Chapter 8 quiz. So there's a number of quizzes here: Chapter 8, Chapter 9, Chapter 10, and Chapter 11. And Chapter 11, so when you look at the content that's not available yet, if you move your mouse over the content isn't available, it tells you what you have to earn. So you have to earn 8 points or higher on quiz number 11 for you to be able to access the content. So I'm going to pause this video now and do the quiz so that I can actually access the content. So give me just a second here. All right, so now that I've gotten 10 out of 10 for Chapter 11 quiz, I can now open these. And what you've got here is you've got questions about blood type inheritance. You've got questions about hemophilia and color blindness. Those are interesting. So blood type inheritance is interesting because it's an inheritance where you have codominant alleles. It's also interesting because it simply affects you as humans. Hemophilia and color blindness are interesting because they are sex-linked inheritance, an example of sex-linked inheritance. Here's the Who's the Daddy assignment, which was, like I said, a real question, a real something that happened to me. The lethal alleles, the book talks about lethal alleles. Lethal alleles are alleles that kill you before it's your time. Why would they stick around if something kills you before it's your time? The missing person, that particular assignment, I have it on a discussion board so that you can help each other out. The hardest thing about this particular assignment, and I'm going to go real quick to go to the assignment materials. In the assignment materials, you'll see, so what you have is you're a detective working a crime scene, and you've got 3 missing persons, and you've just found a crime scene with a whole lot of blood but no body, and you're trying to figure out if one of those 3 missing people could potentially be the victim that was at the crime scene. The things that people struggle with the most is this interpretation right here, of the blood that's found at the crime scene. There's an explanation how to interpret it right here, but the basic gist of it is, so this is one slide. This is the slide where you tested the blood at the crime scene. And on the slide with the test of where you tested the blood at the crime scene, this was pre-incubated. This left side was pre-incubated with anti-A, so it would react with any blood that has A in it. And this side on the right was incubated with anti-B, so it would react with anything that has B in it. This here is a different slide. That's your control slide. Your control slide here shows you what it looks like if it doesn't react. It looks like this. And what it looks like if it does react. So the first thing you have to figure out is which one of those, if any of them, react, and what does that tell you about what kind of blood this is. So that's the thing that trips most people up. So once you get past that, the rest is pretty simple. When you're answering these questions, please note that not all options are possible. I'm not asking you for what are all the possible options for this person, right? What what I'm asking you, given the search, the other information you have, what are the possible genotypes and phenotypes that everybody has? So this is a little more complicated than it looks um, from at first glance. And like I said, I'm using it on a discussion board so that each one of you is, if you get stuck, that you can go, oh, I missed that, but my classmate got this, and now that I read what the argumentation is that my classmate made, now this makes a whole lot more sense to me. So that's that. And then once you have that discussion, then you can go to the who's the victim assignment and get that right. Um, and you have multiple attempts. So if you get it wrong the first time around, you can go back to the discussion board and say, "Hey, I don't quite get this. So what's the deal with this one?" Um, and that's it for this week. So hopefully you have some fun with this. Like I said, isolate DNA with the kids if you've got any. It works much better with strawberry if you happen to have strawberry. If you happen to have anything else, I mean, spit works all the time, but other fruits may work just the same as strawberry. Strawberry just works really well because it has so much DNA inside of it. But that's all I've got for you this week.